probability of pulling out the second one? They're exactly the same. Okay? They're exactly the same. Okay, so what the odds of pulling those 25 letters out, and this thing, there it goes, these and these is the same. And it's very large. The probability of pulling those out is very, I guess it depends on how you look, it could be very small. It's very rare to do that. Okay, so what if you changed a letter in the second one? What if you changed a letter? Would you still have the same meaning there? It would change it. You wouldn't have the same function. This is how proteins are arranged. That's how proteins are arranged. Proteins must be in order to perform a function. So proteins are amino acids that are lined up in a very specific sequence like that second line of letters is. And out of the vast number of proteins that are possible, the proteins show over and over again in your DNA to be very specific and very functional for the purpose that they're there for. Okay, so how do we recognize complex specified information? Probability alone is not enough for complex specified information because those two lines, the probability is the same of pulling out those two lines randomly. It just seems like one is more difficult because it means something to us. But probability is not enough. So improbable, Danny, I'm sorry, one ahead there. Okay, improbable is not enough. We need more than that. And an example would be if you're playing poker. If you're playing poker, it's just as probable to get 4 aces as it is to get a terrible hand. The probability is the same. But what if you started getting 4 aces, 4 or 5 hands in a row? Well, you'd start to think, well, I think the deck is kind of stacked there. There's probably some intelligence or something's going on behind the scenes there. Okay, so unlikely things happen all the time. We don't infer design because something is unlikely. We need more than that. We need specification. We need something if it's specified. And it's specified if it matches a previous pattern. That's what makes it specified. And an example of this would be if you went out to see Mount Rushmore. If you looked at Mount Rushmore, you might say, how did that happen? What caused that? And you'd look at it and you'd go, wind and erosion? You know, maybe because the wind and erosion are affecting it right now. But when you look at it, it matches a previous pattern. It has Teddy Roosevelt, it has Abraham Lincoln. It matches a previous pattern. That's what makes it specified. Next slide, Danny. Oh, I'm sorry, could you back up one? Okay, so specified information has purpose or function. It has control. There are layers of control, systems, codes, irreducible complexity. The only known cause of complex specified information is intelligence. And a flagellar motor is an example in biology of complex specified information and irreducible complexity. Irreducible complexity means that if you take one part, if there's multiple parts, you take one part out and it won't work. A mousetrap, you take one of the parts out, it doesn't work. One slide forward, Danny. Okay, so this is the flagellar motor, and this has been studied endlessly. They know everything about this because one thing, it's so fascinating. This is the little flagella that comes out of the bacterial cell. Okay, and this shows the order of assembly when a cell duplicates. It shows how that is made, and if you look here on the time, it's 20 minutes. The cell will build that in 20 minutes. It shows the order of it too, starting at the bottom. First you have the MS rings and the exporter proteins, and that's the green part down there, and it starts and it's building its way up. There's a stator, there's a rotor, there's a rod, there are L and P rings which seal it like a gasket. Then there's a hook and the junction and a filament. That motor will spin at 6,000 to 17,000 RPMs. without the flagella. Okay, with the flagella, it'll spin 200 to 1,000 RPMs. It has an instantaneous rotational reverse. And the way it does that is it changes very slightly the position of a protein. And that makes the flagella instantly reverse and head in the other direction. So this is irreducibly complex. You take any one of those out and it doesn't work. But according to Darwin theory, it had to gradually be built. It had to have a piece here, and then you'd have a mutation, and another piece would form, then another mutation, another piece would form. Next slide, Danny. So DNA stores the information that, that builds proteins. It's the code. So information is in every cell, but how did it get there? Information in DNA is organized in the same way a computer organizes and stores data. A computer uses a binary code. The DNA uses 4 bases rather than 2. Next slide. So I'm sure a lot of people recognize this. This is the Ascii code for computers, and that shows that like a 1 is represented by, everything's represented by 0s and 1s. And so every number and every letter is represented, and every punctuation mark, and that's how computer code is written. The same happens with DNA. Next. Bill Gates said that DNA is like a computer program, but far more advanced than any software created. Richard Dawkins, who's an atheist, said that the machine code of the genes is uncannily computer-like. Olaf Coopers, who's at the Max Planck Institute in Germany, a very famous top-of-the-line in studies, the problem of the origin of life is clearly, basically equivalent to the problem of the origin of biological information. Hubert Yaki, the genetic code is constructed to confront and solve the problems of communication and recording by the same principles in modern communication and computer codes. He also said the technology of information theory and coding theory has been in place in biology for 3.8 billion years. Okay, we're going to review a little bit. One slide forward. Okay, so the Grand Canyon. If you went out there and looked at that, is that complex specified information in there? Yes, no? Nope, that's wind and erosion, right? Yeah, there's no information behind that. Maybe Shannon information, maybe, if you're talking about little molecules and, you know, when you put one there, it reduces information a little bit, but that's not complex specified information. That's a natural process. It's wind and erosion. There's no intelligence behind that that causes that. Okay? Speed limit sign. Yeah, it's definitely semantic information. It serves a purpose. If you think, you know, you should try to figure these out before you ask the ultimate question, is there intelligence behind it? But yeah, we know. There is an intelligence behind most of them. Okay, let's say you're walking at ISU and you go over a little bridge and you look down and you see ISU spelled out in flowers. Is that complex specified information? Yeah, it would be. It serves a purpose. There's an intelligence behind it. It conveys a message and information. There's a code there because letters are codes. Okay, old pottery. You're an archaeologist. You're digging and you find a pot. Is that complex? Is there an intelligence behind that? Would you say that that just happened naturally? Or would you say that, yeah, you do your studies, you research it, then you go, no, there's somebody built that. There's intelligence behind it that cannot happen from normal, natural processes. Okay, we see complex information everywhere because of the computer age. Computers, cell phones, fax machines, books, automobiles, houses, airplanes, stoplights, on and on and on. Look around the room if there's a book in here. The lights, the computers, those are all intelligently designed. And they go according to the rules. They are improbable to form naturally, specified from a previous pattern, serves a purpose and a function, has control, system, and codes, and sometimes irreducibly complex. Next slide. Okay, so as a review, Information, that's what we're talking about. Recognize it, it requires intelligence. We're talking about complex specified information. Complex specified information is in biological systems. It's improbable, matches a previous pattern, purpose or function, layers of control, irreducible complexity. This requires an intelligence. It requires a designer. Danny? Okay, so let's define evolution. We're defining everything else, let's define evolution. It's a very broad term. Okay, people mean different things by evolution. Even scientists mean different things when they say evolution. Okay, but strictly speaking, Darwin said that we all descend from a common ancestor. He said that you must look at nature only from natural causes, only with a natural cause. So he said that from that common ancestor, there's descent with modification through natural selection. So you had your basic beginning, however, and he never addressed it, but however that supposedly is supposed to get started. And then you have a mutation, and or something changes, and if that survives, then it goes on. If it doesn't, then it dies out. That's descent through modification through natural, or descent with modification through natural selection. Darwin said that his was a very slow, gradual change. So then Mendel comes along, and Mendel actually lived the same time as Darwin too, but they didn't know each other. There's no evidence that those two guys met. He did his pea experiments, if you remember those from high school, and then DNA was discovered in the 1940s and 50s, and so those merged, those theories merged into what's now called neo-Darwinism. And the theory goes, there's a mutation in DNA, then natural selection causes a slow, gradual, random change. That's the current theory of evolution. It merged with the genetics that we learned about. Okay, so selections for traits can only act on immediate benefits from a random mutation. Darwinism cannot look ahead. It can't say 3 generations down the road, we're going to need this, and so we're going to plan it now. It can't do that. It's only immediately. You get the change, if it lives, it goes on, if it dies, it doesn't. That's Darwinism, period. There's nothing else. Okay, slow and random. Okay, change over time. Everybody agrees with this. Even intelligent design people say yes. Nature's going to gradually change over time. Nobody disagrees with that. If you take 2 dogs and you mate them, you're going to get a different dog. If you get a big one and a small one, they're going to be somewhere in between. Nobody disagrees with that. Okay, that is what they call microevolution. Okay, and macroevolution is different. It's an evolution change above the species level. Because when they examine fossils and animals, what this record shows, they won't say this with Darwinism, but this is changing, is you have a species, and then all of a sudden there's a big jump. And then you have species, and all of a sudden there's a big jump. And so that's called macroevolution. Evolution above the species level. This is an area of disagreement. But even in intelligent design, there are some scientists who say that macroevolution is real, and it really does happen, and they have explanations for it. So not everybody disagrees with that, but a lot of ID scientists say macroevolution doesn't happen either. Okay, but the big question that ID says is, does neo-Darwinism have the creative power to provide the complex specified information in nature? Or is it a natural cause? Because microevolution requires very little change in information. Macroevolution requires an enormous amount of new information. Next slide. In The Origin of Species, Darwin said if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. To suppose that the eye, could have been formed by evolution seems, I freely confess, absurd in the highest degree. Charles Darwin said that. He later on went on to try to defend that, but with his theory. But Darwin admitted that if you could find something that could not be made by gradual successions, that his theory would break down. Next slide. Okay. This is a part that for the next 10 minutes. If you don't completely understand this, it's not a big deal to understand the big picture. But I'm going to try to explain how your body makes proteins, okay? And that's what these difficult things are for. Okay, so it is amazing. I think it's important to know if you're gonna learn about this. But if you're like, what the heck's he talking about? That's okay, because in maybe 5 minutes, 10 minutes, I'll let you know when we go to the part that you need to start paying attention on it. Okay? Not that you shouldn't pay attention on this. Okay, but the central dogma in biology is DNA makes RNA, makes protein, makes us. That's what biology says. DNA makes RNA, makes protein, makes us. Next slide. So we're going to see how DNA does this. We've got, I think everybody has seen this picture before. This is the double helix of the DNA. And so the squirrely, the lines, we're not really too concerned about that part. What we're concerned about are the C's and the A's and the T's and the G's in the middle. That's, and you see a little chart on the bottom right-hand side. Adenine, thymine, cytosine, and guanine. Those pair up and match together. So if you have a guanine on one side, you have to have a cytosine on the other, and vice versa. If you have adenine on one side, you have to have thymine on the other, and vice versa. That's just the way that it works. Okay, so one important thing to remember here is that one of the theories of the origin of life is that it just happens because of how chemicals are. Okay, it's just going to naturally form, like ice crystals form when it gets cold. Like salt and chlorine come together for sodium chloride, for salt. You know, that's one of the theories of how the origin of life is. That it's just natural for the molecular processes, how they line up, to fit and to do that. But when they discovered DNA, it just shot that whole theory down. Because if going up the ladder this way, where it says CAG on the side there, my pointer's not working really well. Right there. Going up the ladder this way, there is no chemical bond between the A and the G and the C going up the ladder this way. There is a cross this way, there are hydrogen bonds, but not going up this way. And this is the way that codes for the proteins. It goes up the ladder. And you'll see that more here as we go along. So when they discovered that, they realized, well, that theory is shut down because there's no chemical bond there. How would that naturally come together to form a protein? It can't happen. There's no bond. A way to think of this is, you know those letters that you have on refrigerators that, you know, all kids have and they spell their name on the refrigerator? You put those letters on the refrigerator, there's a chemical, or not a chemical bond, but there's a magnetic bond between the magnet and the refrigerator. Okay? But you spell out 7 years ago, there's no bond between the letters between there. Somebody has to arrange those letters. There's a bond underneath, but not arranging the letters. The 4 didn't come together because of some process. Okay, next slide. So DNA makes proteins. There are 50,000 different proteins in your body as best we know. They don't have a good estimate on this. Some people say there's over 1 million. Proteins are the workhorse of your body. They do everything in your body. Hormones, 50 different hormones. Enzymes, that makes chemical reactions happen faster. 75,000. Muscle, bone, connective tissue, neurotransmitters, all proteins. So proteins are from amino acids. And there are 20 amino acids in your